Okay, so last week we left off talking about what we do with that filtrate after we form it in our renal corpuscle, which involves tubular reabsorption, which is where we remove stuff back from the tubule fluid and bring it back to the blood. And then we're also going to talk about secretion, which is the opposite of that, moving stuff from the blood and into the tubule. Okay, we left off talking about the reabsorption processes that take place in the proximal convoluted tubule, the very beginning part of our renal tubule, which once again is kind of that jumbled up mess right there on the other side of the renal corpuscle. What we're going to talk about now is what happens in the loop of Henle or nephron loop, which is going to see continued reabsorption of water along with some other stuff. But because the proximal convoluted tubule reabsorbs about 65% of our filtered water, fluid that enters the loop of Henle is very different chemically than what it was entering the proximal convoluted tubule. Okay, the loop of Henle will continue this reabsorption of water about 15%, plus the solutes chloride, potassium, calcium, magnesium, and bicarbonates. But for the first time, water is not coupled to the movement of these solutes because part of the loop is relatively impermeable to water. And that part is actually what's highlighted here in green, what we describe as like the thick segment. And that's typically found on the ascending limb, whereas the thin segments are more so in the descending limb, and then a little bit where it kind of U-turns upwards a little bit. The thin segments are very permeable to water, especially the descending limb. Okay, what takes place in the loop of Henle can be described as the countercurrent multiplier mechanism, which refers to the interactions between the ascending and the descending loops of Henle, which operates to increase the concentration on the renal medulla. Essentially, what happens here is twofold. Something is taking place on one side in the ascending limb that affects the descending limb. Okay, and I can kind of just show it best here on the next page, where we can actually see what's going in which direction. I'll start by talking about sodium and chloride, which, as we would commonly call that, is table salt. Okay, in the ascending limb, in those thick segments, we are reabsorbing the salt, and that's because of the pumping action, and that's actually what this figure is showing here, the pumping action of this pump here. Okay, and that's going to create a very hypertonic solution in the renal medulla. If you guys remember the other day, I described the kidneys like if you were to eat them or to like taste the center, it's going to taste super salty, and it's because of what's happening here in this ascending limb. Okay, and the reabsorption of sodium and chloride in the ascending limb, that puts a lot of salt in the interstitial fluid surrounding the tubule. Okay, so essentially all that fluid around the loop of Henle is loaded with salt. Okay, in fact, the deeper in you go into the renal medulla, the more salty it becomes. All these numbers here that you see, notice they get bigger the farther down you go, and then the farther back up, back out into the cortex, the lower the number is. You don't really need to know the numbers, but this numbering is telling you the drive to get water out of the tubule and into that interstitial fluid. If you remember back at the beginning of the semester, when we first introduced the words hypertonic, hypotonic, isotonic, I talked about what happens if you drink salt water. Like if you're stuck in a raft in the ocean after, your plane crashes, whatever, something hopefully none of us experience. But if you drink that salt water in the ocean, it's going to dehydrate you. And that's because all that salt that's in that water ends up in your bloodstream, and your water throughout the body is going to go to your blood to try to dilute that. The same process is what's taking place here. Okay, the loop of Henle in the descending limb loses all that water, and we can see that very nicely here, to try to go out where the salt is in that interstitial space. Okay, so what we're doing is we're reabsorbing water via osmosis in the descending limb because of the interstitial fluid of the renal medulla being very hypertonic. which relates to the sodium and chloride being reabsorbed in the ascending limb. Okay. Now that water that leaves the tubule over here gets picked up pretty immediately by blood vessels known as vasa recta, which are intertwined, and they're not shown here, but they're looping around all of the loop of Henle. We can actually see them right here with their special name labeled. And so the water doesn't just go out into the interstitial space and dilute that salt. It gets picked up into the bloodstream. That's not what this figure is showing, but that's what's happening. And so that just contributes to more of a hypertonic environment. Okay. Now, while that's taking place, we're also seeing urea being picked up by the tubule in both the descending and the ascending limbs in those thin segments. So we can say here urea is being secreted because it's going into the tubule, but that urea is actually coming in from the collecting duct where it was reabsorbed. And so what we're seeing is urea gets moved out of the tubule into the interstitial space, and then it's picked up by the tubule, and then eventually it'll start that process over again. And that's why we call this urea recycling. Okay, and that helps cause a buildup of urea in the renal medulla, okay, and so on and so forth. Main goal of that, though, is to promote more water reabsorption. So that also contributes to the movement of the water out of the tubule. So we call it this countercurrent multiplier mechanism because the movement of sodium chloride out draws the water out. And that's kind of the main takeaway for the loop of Henle. What we're going to do now is talk about what takes place in the distal convoluted tubule. As you can see here, water is reabsorbed there. And we see the same thing, and we'll talk more about that with the collecting duct as well. In fact, here we're going to talk about them all together. And if you didn't know this, or if you need to write it down, it might not be a bad idea. DCT is short for distal convoluted tubule collecting duct, not compact disc, but collecting duct. CD. CD short for collecting duct. All right, now I'm going to go back to this slide and kind of read you what is at the top of that next slide that you're just on. Since the salt that we pumped out here has removed itself from the tubule, the fluid that's now inside of this distal convoluted tubule is very different than it was back here and as it was over here. We can describe it as being very hypotonic, which basically means that it's more diluted. It's got a lot more water compared to other stuff. Okay. Well, the renal medulla, which is once again this segment surrounding the collecting duct and the deeper part of the nephron, it's still very hypertonic here next to the collecting duct, just like it was here in the loop of Henle, because they're all next to each other. Okay. Well, this helps form the osmotic drive for the reabsorption of across the walls of the collecting ducts, reabsorption of water across the walls of the collecting ducts, allowing for some water conservation. Okay, and there are two types of cells, principal cells, or I shouldn't say two types of cells, but there are principal cells here in the two areas, the distal convoluted tubule and the collecting duct, that carry receptors for four special hormones that control how much water and salts are reabsorbed. Okay. Angiotensin II, aldosterone, antidiuretic hormone, and antidiuretic peptide are those four hormones. There's a fifth hormone listed here, but it doesn't have anything to do with water or salt reabsorption. Instead, as you can see, it's associated with the reabsorption of calcium. I'll go ahead and talk about that one. Parathyroid hormone, as we've previously learned, released by the parathyroid glands whenever our blood calcium levels are low, what we call hypocalcemia. Okay. It promotes the reabsorption of calcium. So in other words, we're releasing this when our calcium levels are low. We pull more calcium out of the fluid, out of the tubule and back into the bloodstream to hopefully bring those levels back up. Okay. Well, that's nothing to do with water and salt. So that's why I went ahead and talked about it. Okay. These other ones we've already talked about in this lecture. Earlier in the lecture, we learned about angiotensin II, which we learned reduces glomerular filtration rates in an attempt to help reduce water loss and therefore bring our blood volume back up. Okay. Not only does it do that, but it enhances reabsorption of sodium chloride, salt, and water in the proximal convoluted tubule. But it really doesn't do anything as far as what's in the distal convoluted tubule and collecting duct, not directly anyway, but it does stimulate the release of aldosterone. Aldosterone, once again, is described as being the salt retaining hormone secreted by the adrenal cortex when things like our blood sodium levels fall or potassium levels rise. Its job is to cause retention of sodium chloride and therefore water, which reduces your urine volume and raises your blood pressure. So it promotes the reabsorption of salts and water. Okay. Antidiuretic hormone secreted by the posterior lobe of the pituitary when we're dehydrated or when, for example, our blood osmolarity goes up. Okay. It's actually created by the hypothalamus. If you remember that, the hypothalamus makes the hormones, antidiuretic hormone and oxytocin, but the posterior pituitary is who releases it. And if we're dehydrated, one thing that we're going to try to do is not pee out as much water. And so antidiuretic hormone promotes the reabsorption of water and the return of it to our blood. Okay. The opposite hormone of that hormone is the next one, atrial natriuretic peptide. Remember, secreted by the atrial cells of the heart in response to high blood volumes, or in other words, like high preload or filling pressures. And therefore, an easy way to lower your preload, lower your blood volume, is to excrete more salt and water in your urine. So in this case, this promotes the secretion of salt and water in urine, which reduces your blood volume and therefore your blood pressure. So that's what those hormones do to kind of finalize and control our formation urine. Okay. Table 26.4 just kind of lists those same hormones, gives a little bit more detail and specifics onto what's happening, but I'm not going to draw anything specifically from that. And just here on this next slide is a picture to kind of just emphasize everything that we talked about, plus quite a bit more. We started by talking about the renal corpuscle, where we first form what's called filtrate and pull out a lot of water and a whole bunch of other stuff. Things like ions, glucose, amino acids, creatinine, and uric acid, which then that filtrate travels into the proximal convoluted tubule, where we do a lot of reabsorption, especially of water, but also things like sodium, potassium, glucose, various ions. We reabsorb amino acids and urea, as well as some other ions, but we really didn't talk about all those things. And we didn't talk about really any of these things that we're secreting, with the exception of hydrogen ions. Then you get into the loop of Henle or nephron loop, which is where that countercurrent multiplier mechanism is. What we focused in on there was the reabsorption of water, sodium and chloride, or salt, as they come together. And then we talked about the cycling of urea as it's secreted into the tubule. And then in the distal convoluted tubule and the collecting ducts, really kind of focus in again on water, sodium, and chloride. Together that makes salt. And then we've talked just briefly about calcium thanks to the parathyroid hormone. So I actually skipped quite a bit of stuff. I could have gone into detail and talked about everything that's reabsorbed, everything that's secreted in all those different areas, but I'm not going to do that. It's just too much. And so I'm going to keep it quite a bit simpler than that. to help you guys out. Now, at this point, we've formed the urine, which means we got to get rid of it. And this is finally going to be a point where we get to talk about some other organs of the urinary system, although we're not going to talk too much about them, just briefly for those other organs. Carrying urine down from your kidneys are your ureters and muscular tube that pass dorsal to the bladder and entering them from below. Very similar structure to the esophagus in that they are collapsible muscular tube. They perform peristalsis just like the esophagus does. Gravity is going to play a role, but it's not necessary to get that urine to the bladder. Not much to discuss physiologically with the ureters. Bladder itself is a muscular sac on the floor of the pelvic cavity. It's inferior to the peritoneum, posterior to pubic symphysis. That's all anatomy-based stuff, so not much to describe there. It does have a special layer of muscle that gets a name called the detrusor muscle. It is something that will affect and help with the forcing of urine out of the bladder. We'll talk a little bit more about that in a second. On the floor of the bladder, there's a triangular shaped area formed by the 3 openings, 2 from the ureters and 1 to the urethra, called the trijone. Happens to be a common site of bladder infection, but not much else to discuss here, at least now. We'll talk a little bit more about it here in a second on the next page, along with the urethra, which is the tube that transfers urine out of the body. Just like what we saw with the anus the other day when we talked about digestion, there are 2 sphincters that control the movement of urine out of the body, an internal urethral and an external urethral sphincter. The internal, with the letters IN, are easy to remember, is under involuntary control, and that's because it's made up of smooth muscle, whereas the external is made up of voluntarily controlled skeletal muscle, and we'll talk a little bit more about that here on the next page. Down at the bottom are 2 separate illustrations, one of the male reproductive and urinary structures, one for the females. And that is important because this is going to help transition us into our next lecture. The urethra in a male is not strictly a urinary functioning organ. It is a structure that carries semen and sperm, seminal fluid and sperm, for reproductive purposes. So we'll get into that again in our next lecture, whereas the urethra in a female, strictly urinary in function, and is a completely separate structure from the vagina, despite the fact that those terms kind of get used interchangeably. They're definitely not the same thing. All right, let's talk about peeing. As we've talked about pooping here not too long ago, might as well talk about peeing as well. Just like we've seen here for some common things we talk about. We learned previously things like swallowing and chewing have fancy names, so too does peeing. And this is called micturition or to micturate. Not to be confused with mastication. Micturition means peeing, mastication means chewing. Those sound similar but are very different. This is partially controlled by spinal micturition reflex, where the reflex center is in the sacral regions of your spinal cord. So this is an example where the brain is not actually the reflex center, not in charge. It's down in the sacral part of our spinal cord. As the bladder fills up, that activates stress-sensitive receptors in its wall, resulting in action potentials being sent to that micturition center, triggering a micturition reflex. And that's what we're showing here in number 2. Okay. And that's after the bladder has stretched because it starts to fill up. Okay. At this point, the reflex center will activate parasympathetic neurons to produce 2 things. Okay. Contractions of that detrusor muscle, the muscle in the wall of the bladder that I just mentioned, okay, as well as relaxation of that internal urethral sphincter. Okay. And that's this muscle that would be located right there. which once again is under involuntary control. Okay. At this point, a person would have the urge to urinate, but as long as they are, of that age where they are potty trained, they still retain conscious control over the external urethral sphincter because of signals coming down from the brain, okay, coming down from the cerebral cortex, which can voluntarily contract that external urethral sphincter, just like any other skeletal muscle. We have that voluntary ability to control it, okay, which allows for you to hold your pee. Okay. And this is an example, and we get this in early childhood, where we can delay or essentially kind of suppress a reflex for a limited period of time. And that's a rare example. We can see the same situation with defecation. But as we learned earlier in the semester, when we talked about reflexes, most reflexes are involuntary. We can't suppress them, or it's really hard to. Like you sit on the table, doctor taps on your knee, you kick out, right? You can hold your leg back, but your leg's going to kick out because of that involuntary nature of a reflex. This is an example where you can delay it. And delay is a good word because you can't do it forever. You can't hold it back forever. It's a limited period of time before your bladder is going to get so full that you gotta empty it. Hopefully you can go to a place and that's, a convenient situation. Okay. So when you consciously want to urinate, the micturition center inhibits somatic motor neurons that innervate that external urethral sphincter, allowing it to relax. And then the pee comes out through the urethra to the exterior. Make sense? All right, last thing I want to talk about with our urinary system is how it is involved with regulating pH and therefore maintaining adequate levels of acids and bases in our body. We've talked about this before, but your urinary system is one of the two major systems that is most involved with regulating your pH, the other being the respiratory system that you guys just had your test on. And it is important to keep our acids and bases in check. It is a major homeostatic challenge to keep our hydronic concentration and the pH of our body fluids at an appropriate level, because without maintaining those levels in your acid-base balance, normal cell function wouldn't exist. And that's because proteins prefer a specific pH, and the same goes for temperature, keeping them from denaturing where they lose their shape, they come unwound, and then they lose their function. And we talked a lot about that just a couple of class periods ago with our digestion lecture. Okay? When we talked about denaturing proteins, when we first talked about proteins at the beginning of the semester. Remember, if something's happening in the body, good chance that protein is doing it. And so we want to keep our pH throughout the body where it needs to be. Now, not all the pHs are going to be the same. We've talked about how like the saliva has a pH much higher than in your stomach. And the pH that we're going to be most concerned with is your arterial blood. And we like to see it in the range between 7.35 to 7.45. So like 7.4 is like the magic number that we like to see it at, which makes it slightly basic, just above 7.0 or neutral. So it's a really, really weak base. Okay. The removal of hydrogen ions from our body fluids and its subsequent elimination from the body is dependent upon three major mechanisms: buffer systems, the exhalation of CO2, and the excretion of hydrogen ions from our kidneys. Buffer systems utilize buffers, obviously, hence the name, which can act quickly to temporarily bind to hydrogen ions and remove them from solution, which effectively is going to help raise the pH of the body fluids. But the key here is they don't actually remove the hydrogen ions from the body. Okay. The three major buffer systems that I wanted to talk about are listed here: the protein buffer system, the carbonic acid bicarbonate buffer system, and the phosphate buffer system. Protein buffer system is the most abundant buffer in our intracellular fluid and blood plasma. The amino acids that make up these proteins are capable of releasing hydrogen ions when pH rises. That's going to make pH go down. Okay, and that's because this hydrogen ion can combine with hydroxide, forming water. And the other end of the amino acids are capable of combining with hydrogen ions when pH falls, which is going to help bring it back up. Carbonic acid bicarbonate buffer system is based on the bicarbonate ions HCO3 that act as weak acid or weak bases, and carbonic acid H2CO3 that can act as an acid, obviously since it is an acid. And we've talked quite a bit about that carbonic acid bicarbonate water CO2 stuff and that reaction here in the last couple class periods. The final buffer system is a phosphate buffer system, which utilizes various phosphate ions to regulate acid-base balance in the cytosol. Okay. More importantly, to regulate and to remove hydrogen ions from our body, we breathe and we pee. Okay. Breathing obviously is the exhalation of CO2. By increasing the rate and depth of breathing, that's going to result in more carbon dioxide being exhaled, which lowers the levels of carbonic acids in your blood. Overall, that helps to raise your pH. Okay. Remember that increases in CO2 concentration in the body result in a drop in pH due to the increases in carbonic acid, and that decreases in CO2 concentration result in elevations in pH. Okay. So we can either hypo or hyperventilate, and hopefully you guys all know this because you just had your test on it. Hypoventilation means our breathing rate and depth goes down, so less CO2 is exhaled. I don't know why it's showing up this way, but there should be an arrow just like this right there that results in hydrogen ion concentrations going up. Same thing, another arrow pointing here that results in pH dropping. Hyperventilation means that we breathe more rapidly, more deeply, so more CO2 is exhaled, causing H2 or hydrogen ion concentrations to decrease, which causes pH to go up. Now, this is great at regulating your pH in a very quick manner, because thinking about breathing, we're doing it all the time from the day that we're born to the day that we die. And this is a really quick way of regulating pH. With regards to using your kidneys, it's a much slower mechanism, but it's the only way to eliminate acids other than carbonic acid, where those acids are excreted in your urine. Now that's how we help to try to keep it in balance, but in situations, and it does happen from time to time, we can be thrown out of balance. We've seen these terms before, but we'll talk about them again. Condition known as acidosis is where the body's or the blood's pH drops below 7.35, which can result in depression of the central nervous system through depression of synaptic transmission. So it affects the release of neurotransmitters where neurons meet, which therefore depresses your brain and spinal cord. Ultimately can lead to a person falling into a coma, and if severe enough, possible death. Okay. Alkalosis is the opposite, a condition in which the blood pH rises above 7.45. And a major effect of this is overexcitability in both the central nervous system and peripheral nerves, causing nerve impulses to be conducted repetitively, even when they're not being truly stimulated, which can lead to nervousness, muscle spasms, and even convulsions and death. Okay. So you counter a imbalance, either an alkalosis or an acidosis, by a process known as compensation. Okay, so you compensate. Okay, there's two forms of compensation: respiratory compensation and renal compensation. Respiratory compensation is where we change our breathing rate and depth, so changing our breathing, which is what this figure on the right is showing you, which we've already talked about before. And in doing so, that helps control acid-base balance with the regulation of our blood PCO2. Any increase in blood PCO2 lowers pH, whereas a decrease in blood PCO2 increases it. In our feedback loop on the right, we see some stimulus decreasing your blood's pH, or in other words, we've increased our hydrogen ion concentration. which activates those two types of receptors, the centrals in the brain, the peripherals in your blood vessels that send signals up to your brain into the medulla, where that medullary rhythmicity center is. As a result, output signals travel to your diaphragm and other respiratory muscles. And as a result, we contract them more forcefully and more frequently. So we breathe more often and more deeply. So we blow off more CO2, which brings us back into homeostasis in a negative feedback manner. Okay. Compensation utilizing secretion and reabsorption processes in the kidneys is called renal compensation and is much more important in long-term acid-base balance. It's regulating through the blood bicarbonate concentration by either reducing the rate of bicarbonate excretion, like when our pH decreases, or by increasing the rate of bicarbonate excretion when pH increases. Remember, a bicarbonate ion is a base, and bases are the opposite of acids. So if your pH is lower, we want to reduce the rate of bicarbonate excretion, meaning that we want to lose less bicarbonate in our urine. Bringing that back is going to help elevate your pH. And then if your pH levels are already high, we want to get more bicarbonate out in our urine and therefore excrete more bases to drop your pH. Okay. Now, to summarize, there's actually kind of four major forms of acidosis, alkalosis, two different acidosis conditions, respiratory and metabolic. And then there's two different alkalosis conditions, respiratory and metabolic. These happen fairly frequently, just due to different things, whether it's something simple like, you're hyperventilating because of anxiety, which can trigger you to blow off too much CO2, and that causes respiratory alkalosis. Having diarrhea, where you're, losing bicarbonate ions through that, can trigger metabolic acidosis. When you throw up a whole bunch, you lose the acids from your stomach, which can trigger you to become metabolically alkaline. And whenever you have a respiratory situation, you compensate with the kidneys. Okay? Whereas wherever there's a metabolic acidosis or alkalosis, your compensatory mechanism is a respiratory situation. So you either hyper or hypoventilate. It's a really complex cascade of events. If we had more time, I would actually walk you through this kind of complicated little figure. Basically, where you want to be is right where the letter A is, pH at 7.4, PCO2 is 40, plasma bicarbonate is around 26. Okay, but there are conditions where you have to kind of move up and down and around to try to get that pH back to 7.4. I'm not going to get into it. I really don't want to get into too much. Here we can show just simply how we get hydrogen ions out and into the tubule using the intercalated cells, which are found in your collecting ducts. We pump that hydrogen ion out there just like we were pumping it out. I shouldn't say just like, but we're pumping it out here just like we did in our proximal convoluted tubule. We did talk about that today. And then over on the right, we can kind of just attribute everything as far as causes for acidosis or alkalosis to either accumulation of acids or loss of bases that can trigger pH to drop, or in the case of pH going up, it can be simply a loss of acids and or accumulation of bases. And like I said, if we had more time, I'd kind of walk you through things and whatnot, but as far as this slide goes, you don't really need to worry about anything for the exam. Okay, any questions here?